induced breeding and larval rearing of mudfish or Channa striata. And genotyping of Oreochromis niloticus species and other improved strains of tilapia at BFAR and FFTC towards marker-assisted selection in breeding and production. For more information, kindly contact us at 044-940-7157 or email us at fisheries.biotech@gmail.com.
Webinar etiquette. Be on time. Join the meeting early or 15 minutes before the start of the webinar. Check your internet connection. Connect to a good internet connection to avoid disconnection and listening interruption. Mute your microphone. Always mute yourself when you are not speaking and to avoid unnecessary noises. Ask permission to the host or use the raise hand to the reaction box to unmute. You may also use headphones to avoid interruptions. Chat responsively. For questions, comments, and suggestions, use the chat box and Q&A box. All questions will be answered after the session.
The Department of Agriculture issued Administrative Order No. 21, Series of 2005, which underscores the role of biotechnology as a vital tool for research and development, with great potential for delivering products and technologies that will enhance agricultural productivity and profitability, and eventually improve the well-being of the Filipinos. The Administrative Order No. 6 supersedes AO 21 in providing a strengthened agricultural biotechnology research, development, and extension through a restructured mechanism for the coordination on and implementation of the R&D agenda and program on agricultural biotechnology. The National Fisheries Research and Development Institute, or NFRDI, as a primary research arm of the Bureau of Fisheries and Aquatic Resources, or BFAR, was identified as host to the DA Fisheries Biotechnology Center, or FBC. The establishment of FBC provides an environment conducive for conducting and coordinating fisheries biotechnology R&D and related scientific endeavors. DA-NFRDI FBC is envisioned to be a lead center in all fisheries biotechnology-related endeavors focused in the development and management of fisheries and aquatic resources in the Philippines, with the mission to develop strong linkages, collaborations, and networking among fisheries biotechnology stakeholders towards the promotion, enhancement, and improvement of fisheries biotechnology in the Philippines. Currently, FBC is instituted at the National Fisheries Research and Development Institute and housed at VFAR NFFTC, Silasio Compound, Science City of Muñoz, Nueva Ecija. RDI Fisheries Biotechnology Center spearheads 9 collaborative projects with DA Biotechnology Program Office, BIFAR NFFTC, and other institutions. Development and strengthening the capacity of Fisheries Biotechnology Center in the establishment of Priority Fisheries Biotechnology R&D program. Biotechnological approaches in the production of mud fish or Chana strata using induced spawning hormones. Development and application of molecular-based diagnostic technique as a platform in advanced surveillance and detection of emerging Bangus viral disease syndrome, iridovirus, ranavirus in milkfish or Chanus chanus. Establishment of an advanced diagnostic detection method for tilapia lake virus and other emerging aquatic viruses in the Philippines, surveillance, profiling, and molecular disease diagnostics. Molecular identification of bacterial tilapia diseases and other emerging bacterial pathogens in freshwater fishes. Morphological and molecular identification, culture and breeding evaluation of jungle perch or Culia rupestres in captivity. Development of induced breeding and larval rearing of climbing perch or Anabas testudineus. Refinement of induced breeding and larval rearing of mudfish or Chana strata. and genotyping of Oreochromis niloticus species and other improved strains of tilapia at BIFAR and FFTC towards marker-assisted selection in breeding and production. For more information, kindly contact us at 044-940-7157 or email us at fisheries.biotech@gmail.com.
Webinar etiquette. Be on time. Join the meeting early or 15 minutes before the start of the webinar. Check your internet connection. Connect to a good internet connection to avoid disconnection and listening interruption. Mute your microphone. Always mute yourself when you are not speaking and to avoid unnecessary noises. Ask permission to the host or use the raise hand to the reaction box to unmute. You may also use headphones to avoid interruptions. Chat responsibly. For questions, comments, and suggestions, use the chat box and Q&A box. All questions will be answered after the session.
The Department of Agriculture issued Administrative Order Number 21, Series of 2005, which underscores the role of biotechnology as a vital tool for research and development, with great potential for delivering products and technologies that will enhance agricultural productivity and profitability, and eventually improve the well-being of the Filipinos. The Administrative Order Number 6 supersedes AO 21 in providing a strengthened agricultural biotechnology research, development, and extension through a restructured mechanism for the coordination on and the implementation of the R&D agenda and program on agricultural biotechnology. The National Fisheries Research and Development Institute, or NFRDI, as a primary research arm of the Bureau of Fisheries and Aquatic Resources, or BIFAR, was identified as host to the DA Fisheries Biotechnology Center, or FBC. The establishment of FBC provides an environment conducive for conducting and coordinating fisheries biotechnology R&D and related scientific endeavors. DA-NFRDI FBC is envisioned to be a lead center in all fisheries biotechnology-related endeavors focused in the development and management of fisheries and aquatic resources in the Philippines, with the mission to develop strong linkages, collaborations, and networking among fisheries biotechnology stakeholders towards the promotion, enhancement, and improvement of fisheries biotechnology in the Philippines. Currently, FBC is instituted at the National Fisheries Research and Development Institute and housed at BIFAR and FFTC, CLSU Compound, San Isidro Puños, Nueva Ecija. NFRDI Fisheries Biotechnology Center spearheads 9 collaborative projects with DA Biotechnology Program Office, BIFAR, NFFTC, and other institutions. Development and strengthening the capacity of Fisheries Biotechnology Center in the establishment of Priority Fisheries Biotechnology R&D program. Biotechnological approaches in the production of mud fish or Chana strata using induced spawning hormones. Development and application of molecular-based diagnostic technique as a platform in advanced surveillance and detection of emerging Bangus viral disease syndrome, iridovirus, ranavirus in milkfish or Chanus chanus. Establishment of an advanced diagnostic detection method for tilapia lake virus and other emerging aquatic viruses in the Philippines, surveillance, profiling, and molecular disease diagnostics. Molecular identification of bacterial tilapia diseases and other emerging bacterial pathogens in freshwater fishes. Morphological and molecular identification, culture and breeding evaluation of jungle perch or Culia rupestres in captivity. Development of induced breeding and larval rearing of climbing perch or Anabas testudineus. Refinement of induced breeding and larval rearing of mudfish or Chana strata. And genotyping of Oreochromis niloticus species and other improved strains of tilapia at BIFAR and FFTC towards marker-assisted selection in breeding and production. For more information, kindly contact us at 044-940-7157 or email us at fisheries.biotech@gmail.com.
webinar etiquette. Be on time. Join the meeting early or 15 minutes before the start of the webinar. Check your internet connection. Connect to a good internet connection to avoid disconnection and listening interruption. Mute your microphone. Always mute yourself when you are not speaking and to avoid unnecessary noises. Ask permission to the host or use the raise hand to the reaction box to unmute. You may also use headphones to avoid interruptions. Chat responsibly. For questions, comments, and suggestions, use the chat box and Q&A box. All questions will be answered after the session.
The Department of Agriculture issued Administrative Order Number 21, Series of 2005, which underscores the role of biotechnology as a vital tool for research and development, with great potential for delivering products and technologies that will enhance agricultural productivity and profitability, and eventually improve the well-being of the Filipinos. The Administrative Order Number 6 supersedes AO 21 in providing a strengthened agricultural biotechnology research, development, and extension through a restructured mechanism for the coordination on and implementation of the R&D agenda and program on agricultural biotechnology. The National Fisheries Research and Development Institute, or NFRDI, as a primary research arm of the Bureau of Fisheries and Aquatic Resources, or BIFAR, was identified as host to the DA Fisheries Biotechnology Center, or FBC. The establishment of FBC provides an environment conducive for conducting and coordinating fisheries biotechnology R&D and related scientific endeavors. DA-NFRDI FBC is envisioned to be a lead center in all fisheries biotechnology-related endeavors focused in the development and management of fisheries and aquatic resources in the Philippines, with the mission to develop strong linkages, collaborations, and networking among fisheries biotechnology stakeholders towards the promotion, enhancement, and improvement of fisheries biotechnology in the Philippines. Currently, FBC is instituted at the National Fisheries Research and Development Institute and housed at BIFAR NFFTC, CLSU Compound, San Isidro Puños, Nueva Ecija. NFRDI Fisheries Biotechnology Center spearheads 9 collaborative projects with DA Biotechnology Program Office, BIFAR NFFTC, and other institutions. Development and strengthening the capacity of Fisheries Biotechnology Center in the establishment of Priority Fisheries Biotechnology R&D program. Biotechnological approaches in the production of mud fish or Chana strata using induced spawning hormones. Development and application of molecular-based diagnostic technique as a platform in advanced surveillance and detection of emerging Bangus viral disease syndrome, iridovirus, ranavirus in milkfish or Chanus chanus. Establishment of an advanced diagnostic detection method for tilapia lake virus and other emerging aquatic viruses in the Philippines, surveillance, profiling, and molecular disease diagnostics. Molecular identification of bacterial tilapia diseases and other emerging bacterial pathogens in freshwater fishes. Morphological and molecular identification, culture and breeding evaluation of jungle perch or Culia rupestres in captivity. Development of induced breeding and larval rearing of climbing perch or Anabas testudineus. Refinement of induced breeding and larval rearing of mudfish or Chana strata. And genotyping of Oreochromis niloticus species and other improved strains of tilapia at BFAR and FFTC towards marker-assisted selection in breeding and production. For more information, kindly contact us at 044-940-7157 or email us at fisheries.biotech@gmail.com.
Webinar etiquette. Be on time. join the meeting early or 15 minutes before the start of the webinar. Check your internet connection. Connect to a good internet connection to avoid disconnection and listening interruption. Mute your microphone. Always mute yourself when you are not speaking and to avoid unnecessary noises. Ask permission to the host or use the raise hand to the reaction box to unmute. You may also use headphones to avoid interruptions. Chat responsively. For questions, comments, and suggestions, use the chat box and Q&A box. All questions will be answered after the session.
The Department of Agriculture issued Administrative Order Number 21, Series of 2005, which underscores the role of biotechnology as a vital tool for research and development, with great potential for delivering products and technologies that will enhance agricultural productivity and profitability, and eventually improve the well-being of the Filipinos. The Administrative Order Number 6 supersedes AO 21 in providing a strengthened agricultural biotechnology research, development, and extension through a restructured mechanism for the coordination on and implementation of the R&D agenda and program on agricultural biotechnology. The National Fisheries Research and Development Institute, or NFRDI, as a primary research arm of the Bureau of Fisheries and Aquatic Resources, or BIFAR, was identified as host to the DA Fisheries Biotechnology Center, or FBC. The establishment of FBC provides an environment conducive for conducting and coordinating fisheries biotechnology R&D and related scientific endeavors. DA-NFRDI FBC is envisioned to be a lead center in all fisheries biotechnology-related endeavors focused in the development and management of fisheries and aquatic resources in the Philippines, with the mission to develop strong linkages, collaborations, and networking among fisheries biotechnology stakeholders towards the promotion, enhancement, and improvement of fisheries biotechnology in the Philippines. Currently, FBC is instituted at the National Fisheries Research and Development Institute and housed at BIFAR NFFTC, CLSU Compound, Science City of Muñoz, Nueva Ecija. NFRDI Fisheries Biotechnology Center spearheads 9 collaborative projects with DA Biotechnology Program Office, BIFAR NFFTC, and other institutions. Development and strengthening the capacity of Fisheries Biotechnology Center in the establishment of Priority Fisheries Biotechnology R&D program. Biotechnological approaches in the production of mud fish or Chana strata using induced spawning hormones. Development and application of molecular-based diagnostic technique as a platform in advanced surveillance and detection of emerging Bangus viral disease syndrome, iridovirus, ranavirus in milkfish or Chanus chanus. Establishment of an advanced diagnostic detection method for tilapia lake virus and other emerging aquatic viruses in the Philippines, surveillance, profiling, and molecular disease diagnostics. Molecular identification of bacterial tilapia diseases and other emerging bacterial pathogens in freshwater fishes. Morphological and molecular identification, culture and breeding evaluation of jungle perch or Culia rupestres in captivity. Development of induced breeding and larval rearing of climbing perch or Anabas testudineus. Refinement of induced breeding and larval rearing of mudfish or Chana strata. And genotyping of Oreochromis niloticus species and other improved strains of tilapia at BFAR and FFTC towards marker-assisted selection in breeding and production. For more information, kindly contact us at 044-940-7157 or email us at fisheries.biotech@gmail.com.
Webinar etiquette. Be on time. Join the meeting early or 15 minutes before the start of the webinar. check your internet connection. Connect to a good internet connection to avoid disconnection and listening interruption. Mute your microphone. Always mute yourself when you are not speaking and to avoid unnecessary noises. Ask permission to the host or use the raise hand to the reaction box to unmute. You may also use headphones to avoid interruptions. Chat responsively. For questions, comments, and suggestions, use the chat box and Q&A box. All questions will be answered after the session.
The Department of Agriculture issued Administrative Order Number 21, Series of 2005, which underscores the role of biotechnology as a vital tool for research and development, with great potential for delivering products and technologies that will enhance agricultural productivity and profitability, and eventually improve the well-being of the Filipinos. The Administrative Order Number 6 supersedes AO 21 in providing a strengthened agricultural biotechnology research, development, and extension through a restructured mechanism for the coordination on and implementation of the R&D agenda and program on agricultural biotechnology. The National Fisheries Research and Development Institute, or NFRDI, as a primary research arm of the Bureau of Fisheries and Aquatic Resources, or BIFAR, was identified as host to the DA Fisheries Biotechnology Center, or FBC. The establishment of FBC provides an environment conducive for conducting and coordinating fisheries biotechnology R&D and related scientific endeavors. DA-NFRDI FBC is envisioned to be a lead center in all fisheries biotechnology-related endeavors focused in the development and management of fisheries and aquatic resources in the Philippines, with the mission to develop strong linkages, collaborations, and networking among fisheries biotechnology stakeholders towards the promotion, enhancement, and improvement of fisheries biotechnology in the Philippines. Currently, FBC is instituted at the National Fisheries Research and Development Institute and housed at BIFAR NFFTC, CLSU Compound, Science City of Muñoz, Nueva.
Good morning, everyone. Welcome to the second day of the DA-NFRDI FBC's 5th National Fisheries Biotechnology Symposium.
Recording in
To
progress.
officially start this activity, let us all welcome our hosts and moderators, Dr. Benjamin Joseph Bumanlag and Ms. Angeli A. Hente, Science Research Specialist 1 of the DA-NFRDI FBC.
Once again, welcome everyone to our second day of our plenary session. Good morning, Doc Benj.
Good morning, Ms. Angeli, and to our participants here in Zoom and in our live stream on YouTube and Facebook Live. This is the second day of our symposium. We really had a great and very insightful plenary on our first day. Ms. Angeli, what was your favorite part from yesterday's presentations?
I was particularly interested with Sir Spalma's work on oral delivery of FSH and GnRH, which evidently advanced in the maturation of sex-changing grouper, and of course, with the findings of Dr. Lumaino on the isolated isoforms of GnRH and its receptor in Chambackerel with very promising studies. I would say that technology will apply in biotech research here in our country and has really taken a bigger leap, and we will more that out of today. So how about you, Doc Benj?
For me, I like the presentation of Dr. Ozdemir about chromosome manipulation techniques, how it's performed, and its applications on modern aquaculture. So yesterday was filled with informative and engaging lectures, so I'm pretty sure our participants are excited because we have a new set of interesting topics to cover for today.
At this moment, we would like to thank for the support of DA Biotechnology Program Office, DA DFAR National Freshwater Fisheries Technology Center, and DA National Fisheries Research and Development Institute Fisheries Biotechnology Center for making this symposium possible.
As of now, we already have 117 participants from the Zoom platform, and a lot more coming from our Facebook and YouTube live stream.
Follow us at fish.biotech in our Facebook page and visit our website at fbc.nfrdi.da.gov.ph for more updates on our activities here at NFRDI FBC. For those having difficulties in connecting in our platform, kindly refresh your device, and those who are already connected, just sit back, relax, and enjoy the rest of the event.
To start with, allow me to introduce our first presenter. She is the Chief Science Research Specialist of the National Fisheries Research and Development Institute, Freshwater Fisheries Research and Development Center. She earned her Doctor of Philosophy degree in Environmental Science at the University of the Philippines at Los Baños, Laguna, and specializes in fish biology, breeding and culture, inland fisheries, and aquatic resource assessment, community development, and management. She has done several studies and published papers on Lake Taal and Sardinella Tawilis. She has also received several scientific awards and recognitions. One of the most significant contributions of her research was the declaration of closed fishing season and establishment of Tawilis reserve areas in the lake. She is the focal person of NFRDI National Stock Assessment Program for Freshwater, the program leader of the NFRDI Taal Lake and Tawilis Research and Conservation Program, and a member of the Al Volcano Protected Landscape Research Council. Her advocacy continues for the conservation and management of the only freshwater sardinella in the world, the pride of Batangas and the entire country as well. Let's all welcome Dr. Maria Teresa Marcena Mucha on her presentation titled Induced Breeding Using Human Chorionic Gonadotropin and Luteinizing Hormone-Releasing Hormone Analogs in the Reproduction of Caranx ignobilis. Let us give her a virtual round of applause.
Hello, good morning, everyone.
Good morning, ma'am.
Yeah. Is
Morning,
my
Doc.
slide okay? Slide presentation is okay already. Yeah. Okay, to
Yes,
all our guests and our viewers, our plenary speakers also, good morning to everyone. First of all, I would like to thank the Fisheries Biotechnology Center headed by Dr. Castro Resca and Dr. Jodis Danting of BIFAR and FFTC for inviting me here in the 5th National Fisheries Biotechnology Symposium. Thank you also for giving me this opportunity to share to everyone what NFRDI has been doing in developing the breeding and culture technology of Caranx ignobilis and Malipto. Malipto is also one of the pride of Taal Lake aside from Tawili. So our center in Batangas is also focusing on the research and development of this very high value and important species. This morning, I will present my topic is about the induced breeding using human chorionic gonadotropin or HCG and the luteinizing releasing hormone analog or LHRHA in the reproduction of Caranx ignobilis. For my outline, I will discuss some information on the biology of Malipoto and some aspects or concepts on induced spawning or induced breeding, although it was discussed very extensively yesterday by our very good speakers. I'll just give you some background on the induced breeding or what we did with Maliputo. Next is the NFRDI, Freshwater Fisheries Research and Development Center Breeding Program on Maliputo, specifically on broodstock development and development of protocols on induced breeding of Maliputo using the two hormones, the HCG and the LHRHA. And lastly, some ways forward. Okay, Caranx ignobilis is also called, the English name of this is giant trevally. It is also known as the jack, pompano, and the giant kingfish or uluwa. Is a species of large marine fish classified in the jack family Carangidae. It is locally known as talakitok, maliputo, or mamsa. It is the largest fish in the genus Caranx, growing to a maximum of size 170 centimeters, and it has a weight as much as 80 kilograms. The giant trevally inhabits a wide range of marine, brackish, and freshwater environments. And it is widely distributed in the Indo-Pacific region throughout the tropical waters with a range stretching from South Africa to Hawaii, Japan, and Australia. Before I continue, I have a trivia for you. Okay? Do you know what fish you can find in our 50 peso bill? Oh, you can find there in my presentation the 50 peso bill. Yes, the species that you can find or the fish that you can find in our 50 peso bill is the Caranx ignobilis or maliputo. I think it was featured by Bangko Sentral in their celebration of centennial anniversary. This design was made during their anniversary because Caranx ignobilis or maliputo is also one of the pride of Taal Lake. So maliputo is considered as a high-value species. Its price range from 130 to 500 pesos per kilo. It is a commercially important fish in capture fisheries, and it's caught mainly by gill net, ring net, beach seine, and fish coral. And it's the major regions that catch this species in the capture as well as in the culture environment are from the regions 2, specifically in Cagayan, Region 6, Iloilo, and Region 3, Central Luzon. It is also reared in cages and ponds in Lake Taal and Balayan Bay. From our latest inventory, we have enumerated at least 100 cage owners which we are helping also at this time for their grow out culture of maliputo in cages. Maliputo is a good aquaculture species. However, the limitation there is the limited supply of fingerlings because most of them are caught from the wild. And this is one of the reasons why NFRDI conducted this study or this research and development program because we want to increase the production of maliputo being a very good candidate for aquaculture. Okay, before I discuss the results or the studies of NFRDI, I would just like to give some information on what is the meaning of induced breeding and how it is being used to propagate fin fishes like Caranx ignobilis. So by definition, Induced breeding is a technique where fish breeders are stimulated by hormone introduction to breed the fish in captivity. It is also sometimes called as an artificial breeding. And hypophysation is a technique that employs the injection of the pituitary gland extract or various gonadotropin hormones in the body of the fish, which is needed to breed for breeding purposes. So in induced breeding, we also take into consideration some environmental factors because these affect the reproductive cycle of our fish, like the photoperiod, water temperature, flooding and water current, tides and cycles of moon, weather cycles, spawning substrate, nutrition, disease and parasites, presence of other fish, and hormones that produced by the endocrine system, which controls the reproduction cycle or reproduction or production of our hormones. So these are important environmental factors because when we are doing our induced breeding of Maliputo, we do not only rely on the hormones that we inject to our fish, but also we documented the water temperature, the even the phase of the moon. We try to document when is the best time that we will inject the fish. So all of these are very important environmental factors to consider for a successful induced breeding. So what are the common hormones that are being used for induced spawning? So we have the releasing hormones or the GnRH analogs. These are the mammalian GnRHA or the LHRHA, which are derived from GnRH molecules of mammals. There are also salmon GnRHA derived from salmon GnRH molecules. And the gonadotropins, the most common are the crude fish pituitary extract that comes from the carp or catfish pituitary glands. And then the salmon gonadotropins, mammalian gonadotropins such as the human chorionic gonadotropin. So most of these are already commercially available. And we are using for Karang signobilis or Maliputo, we have already tested the efficacy of using LHRHA, but also the HCG, but with the time that we spend in induced spawning of Maliputo, we tested that the most effective is the LHRHA. Okay. So what is the role of gonadotropins in induced breeding? This is a picture of our endocrine system of a fish. The fish pituitary of the brain below the hypothalamus produces and stores gonadotropin hormones or the GTH, which play a decisive role in the ovulation and spermiation of fish breeders. So these gonadotropins are released under the control of the GnRH or the gonadotropin releasing hormone. Examples of the gonadotropins that are very vital in induced spawning of our fish are the luteinizing hormone and the chorionic gonadotropins. So when we inject our fish, like for the LHRHA, which is a GnRH, cause the pituitary gland to secrete the GTH or the gonadotropin hormone, specifically the luteinizing hormone. Okay, which is very vital in inducing the maturation, the ovulation, and the spawning of our fish. While the injection of HCG increased the level of gonadotropins. So it's already a gonadotropin, so it increase the level of gonadotropin hormone in the fish, which is also necessary to induce the maturation, ovulation, and spawning of our fish. Okay, so in NFRDI, FFRDC, we started our breeding program in 2006. Matagal na, no? When we are starting that, it was just an accidental breeding. So we were breeding that time several freshwater ornamental fish that are bred on that are induced that do not breed in naturally. So we are breeding them through artificial breeding. But when we are trying to dispose these breeders, Maliputo breeders, because we will be focusing on freshwater, we just tried to inject the breeders, the Maliputo. But surprisingly, it spawned. So I think that was our signal. Why not breed Maliputo in captivity that time? But of course, our center was not prepared for that because we are a freshwater center hatchery. We have freshwater hatchery, so we still have to develop our marine hatchery that time. And one of our limited or constraints that time or problem was we cannot find any breeding protocol for specifically for Maliputo or Karangsing nobili. So we develop our own protocol for this kind of species. That's why we can say that this is a first in Philippine aquaculture, a scientific breakthrough which we are still continuing to refine the protocols, and now we are looking forward to its commercialization in the future. So these are our milestones. Now you see we started in 2006, 2008, preliminary breeding trials. And we start to develop our hatchery that time. It took us some time to develop our hatchery. And then our first offspring was in 2009. And then we tried to improve our fertilization, hatching rate of our hatching rates. And then in 2015, we had our first larval dispersal. We tried to improve our broodstock diet. And we tried to refine further our larval rearing. We already conducted our national trainings. And currently, we are verifying the hatchery, larval rearing, and grow out techniques to our cooperators, to specifically private cooperators. And we are getting good results of our breeding program as well as the grow out culture of Maliputo. So this is the area where we conducted our breeding program. We have very limited space. So, and during that time that we started, it is located in Botong Taal, Batangas. Its area is 8,500.5 square meters. So it is, we started with no tanks at all. So we asked for funding to construct our tanks that time. Okay, for the protocol that we have developed and still refining, we start with the broodstock development, okay? So everything starts with good breeders when we are trying to breed our fish. So we collect, when we found out that we can breed it in captivity, because before we just rear them for grow out culture in freshwater marine environment. But when we found out that it can be bred in captivity, we already start collecting our breeders. We start with the fingerling size, which we collect in Balayan Bay. It's just adjacent our center. So we started in 2005, the collection. And we waited for 3 to 4 years for them to be sexually matured. So it took some time for them to use them, because it takes 3 to 4 years to be sexually matured. So during that time, we give them a good diet. When they are sexually matured and we are trying to condition them already for our breeding, so we give them a high protein diet. We incorporate the shrimp, squid, liver, and vitamins so that they will have a good maturation of their eggs, and they have good condition in breeding, and they are healthy, and they will produce more eggs, more good quality eggs. And of course, we maintain good quality of our water all the time. It's a must because they like clear water, because they are carnivorous. They do not want the algae or the aquatic macrophytes, so we have them removed from our ponds. And always replenish our water every week, every day, or every other day. It depends on the water quality. So we use after they are already 3 years or 4 years, we condition them, and we use the luteinizing hormone and con- human chorionic gonadotropin that time. But as we tested our different hormones, what is most viable for us is the LHRHA. And we tried to document its spawning performance, the ovulation, hatching period, fertilization, and hatching rates. So these are the things that we do. We prepare our hormones, then we sedate. We just like the other marine for a while. Just like in other breeding of our or injection of our marine species, we anesthetize them using MS-222. So it took us a while to really get the technique. See, the technique, you cannot find them in books. So it's more on many trial and errors in doing this. So we tried several strategies. But I can say for now that we have already mastered the sedation technique of our malipoto. And also, why do we sedate? It's for the communulation. We want to check the development of eggs, if they are already matured, if they are already good for induced breeding. And then after we selected our breeders, we only select the very good spawners, wherein the eggs must be around more than 0.4 millimeters in diameter, and they are at least 60% of them are big. The diameter is good, diameter is big, and the color of the eggs, we also take a look at the color of the eggs. And for the for the male, the milk should be very milky white. So those are the indicators of which breeders are we going to inject during our cannulation. So our ratio for breeding our malipoto is 2 male and 1 female. And then after that, we place them in our hatchery tanks, in concrete tanks. So we let them spawn there naturally. So we inject them with these hormones, and then we let them after several hours to ovulate and to spawn naturally. Okay. So we get the ovulation period, the hatching period, the fertilization rate, and the hatching rates. And then we document the eggs, the embryonic development, the larval development. So every time, we do this every year because there is a specific season only for malipoto. For the year, we allot the March and April for the spawning or the induced breeding activities for malipoto in our center. That is the best time for us to do the activity. So what what are the results for the past years that we have been doing this? So for LHRHA, uh, and HCG, the mean weight breeders that we usually use is about 5 kilo per fish. So what we obtained during that time is for the LHRHSHA, we have a mean number of eggs spawned per kilogram breeder. This is 223,068. So if you multiply it by 5, it is around 1 million per breeder, right? So this is higher than what we obtained from the HCG. And also the spawning period is for LH, it is 24 to 52 hours, which is longer than the HCG. HCG is 30 to 36 hours. And the hatching period is about 11, they are almost the same. 11 to 17 for LH and 11 to 13 hours for the HCG. So these are the fertilization and the hatching rates. So we can see the LH has higher fertilization and hatching rates. So we can say fertilization has statistically significantly higher than the LH than the HCG. But the hatching rate, this is not statistically significant, but we can see it's comparable. It is numerically higher than the HCG. Okay, for the spawning of eggs and spawned eggs and hatch larvae, we can see that LHRHA, the spawn eggs, is a little bit lower than the HCG, but the hatch larvae is higher for the LHRHA than the HCG. The mean spawn eggs and the mean larvae produced were not statistically different from each other, but we can see that the hatch larvae is more important to us because this will be our focus to how in the next run that we will rear them to fingerlings. So we get a higher hatch larvae from the LHRHE. So we also documented the water parameters. These are we have to consider this very strictly. We have to, the optimum range for temperature should be 27 to 30. And the salinity is 28 to 30. Dissolved oxygen, of course, should be higher than 5, and the pH of 8 to 8.5. So these are just our documentation of our embryo development. We have already documented the whole development from the first cell division after 1 hour to hatching after 11 or 13 to 17 hours. So we have a full documentation of this embryo development for Caranx ignobilis. And for larval development, we also documented this. This is from yolk sac stage to fry stage after 30 days. Okay. Okay, so what are our ways forward? Of course, it's not yet perfect. We still need what I can say is the hatchery, the spawning is already perfect with that, but the larval rearing, we still have to increase our larval survival. So we need to refine our protocols. And very recently, we were able to discover the missing link. So we have to still verify for the larval rearing because we already asked help from the experts expert private hatcheries of milkfish. And they are also saying the same, that it's really very hard to rear for the larval rearing. So we are trying to refine it again. And then at the same time, we are already verifying our protocols, verifying our studies on hatchery, grow out, and nursery with the cooperation of our BIFAR counterpart collaborators and the private fisher folk. Okay, this year and next year we'll have a project on the technology verification of grow out culture of Malipoto, wherein we have identified 30 cooperators or cooperator beneficiaries. Most of them are here in Lemery, Batangas, and Taal, wherein they are already into the grow out culture of Malipoto in cages. And also, if we'll be able to produce more of these karangs or Malipoto, will be on the stock enhancement program of our marine and inland bodies of water. In fact, during our first produce, the initial and then the succeeding produce, we are stocking them already in our fresh and marine bodies of water. Okay, so that ends my presentation, but I have prepared to you a 4-minute video just to share with you our research undertakings on Malipoto. Thank you very much for listening, and I hope you will enjoy our video.
Dito sa Botong Taal, Batangas, sinimulan ng Freshwater Fisheries Research and Development Center o FFRTC ang pagbibreed sa Maliputo noong taong 2006. Sa pamamagitan ng artificial breeding o pag-induce ng spawning nito, naging matagumpay ang pagpapaanak sa Maliputo sa unang pagkakataon. Dahil dito, nabigyan ng pagkakataon ang FFRDC develop ang teknolohiya ng pagbibreed ng Maliputo. Ang Maliputo o Caranx ignobilis ay isang high-value species na may malaking potential for aquaculture. Sa kasalukuyan, nakakaproduce na ng higit 1 milyong similya ng Maliputo ang FFRDC sa isang taon. Ang pagbibreed sa Maliputo ay nagsisimula sa masusing pagpili ng mga ready-to-spawn breeders. Ito ay ginagawa sa pamamagitan ng cannulation. Isa tong paraan ng paggamit ng plastic tube upang makakuha ng sample ng itlog sa mga babaeng breeders at milk para sa lalaking breeders. Ang mga nakuwang sample ng itlog ay ine-examine sa microscope upang malaman ang diameter ng mga ito. Kapag ang mga itlog ay may sukat na 0.4 mm pataas, ibig sabihin ay ready to spawn na sila. Sa mga lalaking breeders naman, ang mga breeders na may milky white na milk ang siyang pinipiling mga spawners. Sa pagpapaanak ng Maliputo, importante ang tamang handling ng mga breeders. Kailangan may iwasan na may stress ang isda habang isinasagawa ang cannulation. Kapag nakapili na ng mga babae at lalaking breeders, sinusukat naman ang haba at timbang nito upang malaman kung gaano karaming hormone ang ituturok sa isda. Pagkatapos ng isang araw na pagkondisyon sa mga napiling isda sa pond, tinuturukan ng hormone ang mga ito sa umaga at sa hapon. Pagkatapos maturukan, dadalhin ang mga breeders sa spawning tanks. Hihintayin na mag-spawn ang mga isda sa loob ng 30 oras. Kapag nag-spawn na sila, kinokolekta ang mga itlog usually tuwing umaga. Pagkalipas ng 16 na oras, mapipisa na ang mga itlog. Pagkatapos mapisa, nilalagay ang mga ito sa larval rearing tanks upang palakihin. Sa simula, pinapakain ang mga larvae ng mga natural food gaya ng rotifer at phytoplankton. Pagkalipas ng 1 linggo, binibigyan na sila ng brine shrimp at coffee pod. Pagkalipas ng 21 araw, pwede na silang i-stock sa mga pond for grow-out culture. Ang teknolohiya ng pagbibreed ng maliputo ay kauna-unahang na-develop dito sa FFRDC. Sa pamamagitan ng teknolohiyang ito, higit na marami ang pwedeng masuplayan ng mga similya ng maliputo, lalo na sa mga interesadong mag-alaga ng isdang ito.
Thank you, Dr. Mucha, for sharing your presentation and initiatives on the induced breeding of Maliputo Caranx ignobilis.
At this point, allow me to introduce our second speaker. He is an alumnus of MSU Maguindanao, where he finished his Bachelor of Science in Fisheries as a Department of Agrarian Reform Scholar in 2007 with distinctions cum laude and University Leadership Awardee. He took his Master of Science in Aquaculture at Central Luzon State University as DOST Scholar. In 2017, he earned his Doctor of Philosophy in Fisheries and Allied Aquacultures at the Auburn University in Alabama, United States as Fulbright Scholar. He is the first Filipino recipient of the prestigious H.S. Wingle Award for Outstanding Doctoral Student, along with other merit-based awards. He authored and co-authored scientific articles published in various peer-reviewed journals. At present, he is an Associate Professor 5 and Vice Chancellor for the Research, Extension, and Development in Mindanao State University, Maguindanao, and is currently President of the Junior Chamber International Philippines Cotabato Chapter. To give his presentation and production of hybrid catfish through xenogenesis, let us welcome Dr. Ramji Y. Udin. Let us give him a warm virtual applause.
Hi, everyone. Good morning. My warmest greetings from MSU Maguindanao and from the province of Maguindanao in the BARM. Allow me to share to you my presentation first. Thank you, by the way, Angeli, for the introduction. Am I audible enough? Thank you.
Yes, sir. Yes.
Thank you. Let me maneuver this. Sandali lang at hindi ko ma-control yung let me stop it. All right. Sandali lang at ayaw hold on for a bit at Ayaw pa rin. Are you seeing it now?
Sir, hindi po pa namin nakikita.
Okay. Dali lang. Okay, what about now?
Yes, sir. Yes, sir.
Okay, thank you. That's it. Anyways, again, good morning to all of you. I am Ramji Odin from the Mindanao State University, Maguindanao. I'm very, very happy and overwhelmed to have been invited by Dr. Cass to speak before all of you today on one of the studies I conducted back in the days when I had my PhD at Auburn University as Fulbright Scholar. And this research is about the production of hybrid catfish through xenogenesis. So I believe that if you're going to search for the thing that you would find most likely is about the movie of James Cameron, Xenogenesis, that was in 1978, but that's not what it is. Basically, just to laymanize it and for everyone to be interested of what xenogenesis is, it's more likely we can compare it to human surrogacy. But before that, allow me to introduce to you what hybrid catfish is. So basically, catfish, this research was conducted in the US when I was doing my PhD at Auburn University. And so my research focused on the hybrid catfish in the US. Catfish are popular food in the southern United States. And the two most commonly eaten farm-raised species in US are the channel and the blue catfish, which has become a staple food. But before, the catfish industry used to grow rapidly as the largest contributor of the US aquaculture production. However, more recently, in the past year, the industry of catfish has stagnated. The production has declined, and thus causing great losses in the catfish industry and to the catfish farmers in the US. This decline in the production has been attributed to the production inefficiencies as well as the susceptibility of the catfish to various diseases, and as well as the continued competition with the imported cheap fish coming from the Southeast Asia. Now, in order to keep pace with those challenges, and as well as improve the efficiency of production of catfish, of both the channel and the blue catfish, what they did there is to cross the channel and the blue catfish to produce the hybrid catfish or their hybrid offspring, which has been found and proved to have a superior performance than the channel catfish and the blue parent catfish. So with the hybrid catfish, they have improved and faster growth. They have better FCR. They also have better resistance to diseases. They can tolerate low oxygen. They can also tolerate overcrowding, stress, and they have more uniform size and shape. And they also have higher dress out percentage and fillet yield. And I think one of the best attributes that they also have, the hybrid catfish has, is the increased harvestability or seinability. They can easily be seined. And so they're like super catfish, no? So compared to their parent channel catfish and the blue catfish. So, however, the thing with the hybrid catfish is, the conventional production of hybrid catfish is usually done on, artificial fertilization. So what usually happens is, since the blue catfish and the channel catfish are not sexually compatible producing hybrid catfish, and therefore the production of hybrid catfish is definitely, not easy. And so with that, it's labor intensive and it requires the sacrifice of the male blue catfish. And imagine the hybrid catfish, the catfish being a huge industry in the US. Imagine how many thousands of blue catfish are being sacrificed every year just to produce the hybrid catfish. And aside from that, in order to produce the hybrid catfish using the artificial fertilization, you need to sacrifice the blue catfish and then get the sperm and then mix it with the eggs of the channel catfish. And then the thing is, they might run out of the blue catfish. Another concern there is the production of the blue catfish is also difficult. The easiest way to produce is the channel catfish, but the thing with the channel catfish is they're more susceptible to diseases. Although they grow faster, but they're more susceptible to grow to diseases. The good thing about the blue catfish is they're more resistant to diseases. So you can see there that the hybrid catfish was able to get the best of both species in terms of their performance. And so now, to deal with that challenge, we make use of xenogenesis technology. Xenogenesis, if we're going to define it based on the definition of my advisor, Dr. Donham, in his book as well, xenogenesis is defined as the production of offspring that are markedly different from either of the parents. So here, we make use of xenogenesis technology where we took the stem cells from the blue catfish and then injected them into the sterile channel catfish and allowed the stem cell to develop into a reproductive organ inside the xenogenic catfish. So here, what we did is we isolated and purified the stem cells with density gradient centrifugation. And then, but before that, after isolating that, before we inject the stem cells to the xenogenic catfish, what we did is we make sure that we manipulate the chromosomes of the normal channel catfish to make it a triploid catfish. So we make use of we produce the triploid catfish using the hydrostatic pressure at around 4,000 to 6,000 psi, and then we injected the isolated and purified stem cells. Specifically, we make use of the spermatogonia stem cell A, and injected it to the triploid catfish. And then we wait until the xenogenic catfish was confirmed to be carrying that foreign material in their reproductive organ. So in human, this is like surrogacy. Now, if we're going to make the xenogenic channel with another normal channel, then supposedly we would be expecting production of a hybrid catfish. So the transplant of the stem cells to triploid channel catfish was then this way. The stem cells from the testes of a 2-year-old juvenile blue catfish was arbored from the blue catfish, and then before we injected the stem cells, we made sure that we have injected it to a confirmed triploid catfish, so triploid channel catfish. In that way, when we manipulate the chromosomes of the channel catfish and make it into a triploid, then they become sterile, and then that way their native reproductive organ to produce a channel offspring or gamete would be suppressed, and thereby allowing the foreign stem cells coming from the other species, the blue catfish, to grow inside the triploid channel catfish. So here, the transfer of the stem cells was done at 3 life stages. So blastula injection, that is 12 hours after fertilization of the channel catfish. And then we also injected the stem cells at a fry stage, that is 1 day after hatching. And then the other stage is the sub-adult injection, where we injected through catheterization the stem cells to a sub-adult triploid catfish. The xenogenic catfish were then allowed to grow in ponds for 3 years until they reach sexual maturity. And then the putative xenogenic channel catfish males harboring the sperm of a blue catfish. So imagine a channel catfish that is confirmed triploid carrying a sperm of a blue catfish were paired and mated to a normal channel female in aquaria following luteinizing hormone injection of both fish at a dosage rate of 100 milligram per kilogram of fish. So now this is the summary of our hybrid spawns produced semi-naturally using xenogenic channel. So one xenogenic male from the sub-adult treatment successfully spawned twice, but produced only 3 and 21 progeny respectively. Now the putative xenogenic fish from the blastula treatment here have yet to produce a viable spawn, but the 3 xenogenic males from the fry treatment produce hybrid spawns with high fertility. Now, one paired twice to 2 different channel females resulted in 20% fertility of the egg mass, and the other 2 males spawned, and the resulting egg masses were 100% fertile. Now, what we did with the samples is we run a PCR on the progeny produced from the semi-natural spawning of putative xenogenic channel males paired to normal channel females. The PCR results confirm the genotype to be that of the channel catfish and blue catfish hybrids. So basically, when we check the PCR results, we were able to find that the hybrid progeny, the progeny of the xenogenic channel catfish crossed with the normal female channel catfish is hybrid. So therefore, the production of xenogenic broodstock must be improved for commercial scale production of broodstock. This is actually the first report of a successful hybrid production of hybrid catfish using the xenogenesis. Now, in other research, basically, I believe in Japan, they have been conducting xenogenesis as well with other species. They have also been successful in using the xenogenesis with other species aside from catfish, and as well as with with other genus and other families. So you can see here the potential of xenogenesis as in improving our aquaculture production. And aside from that, the thing with xenogenesis is that we are actually looking into potentially resurrecting some of the threatened species and some other reproductively difficult to spawn species. So there's really a great promise with xenogenesis. Now, the most important question here is why this research matter to all of us. As we know that the fish is the cheapest source of raw protein, which is vital to human health. And as the human population increases, so as the demand for food. Now we need to produce technologies that can really ensure food security. And in this case, the fish, all other fish that we have, catfish as a staple food for all of us. In the case of the Philippines, tilapia, milkfish, and other important aquaculture commodities being important to the Philippines, we have to make the production sustainable. And this can actually be done through the application of xenogenesis. And with that, thank you so much, everyone.
Thank you, Dr. Udin, for your presentation of the technology that produced hybrid catfish fry through mating of xenogenic males with normal channel catfish. By that, it lessens the sacrifices of our male catfish. Thank you. Once again, thank you, sir.
To proceed, let me introduce our last speaker for this symposium. He earned his Doctor of Philosophy degree in Fisheries Management in 1974 from Auburn University in Alabama, USA, where he conducted his dissertation on the hormonal sex reversal of the blue tilapia, Oreochromis aureus. He has served as Dean of the College of Inland Fisheries and Assistant Director of the Freshwater Aquaculture Center of the Central Luzon State University. In 1988 to 2009, he was the Executive Director of the Philippine Council for Aquatic and Marine Research and Development of the Department of Science and Technology. For his outstanding contributions to Philippine aquaculture, he was conferred the Presidential Rizal Pro Patria, one of the 10 outstanding young men of the Philippines, and IBM Philippines Award for Science and Technology. He has written more than 10 books on tilapia farming, aquaculture, and vermiculture. He has published over 60 scientific papers in international and national journals. Currently, he is an aquaculture or vermiculture specialist for aquatic biosystems, a consultancy and marketing firm in Laguna. He is also an academician of the National Academy of Science and Technology. He is the father of tilapia sex reversal in the Philippines. It is an honor to hear his presentation on the application of tilapia sex reversal technology. Let us all welcome Dr. Rafael T. Guerrero III. Excuse me, Dr. Guerrero, your microphone is muted po.
I'm sorry. Okay. Okay, I'll start with my slides. Okay, did you see? Can you see that? Can you make it bigger, please? Okay.
Not yet, sir.
Is that okay? Okay ba? Okay.
Hindi pa po namin makita,
Yeah.
sir.
Sorry, pinasok ko na eh. Oh, I'm sorry, I did not press something yet, no? Yun, okay ba? Yan. Is it okay
Hindi pa
now?
po namin, hindi pa po, sir.
Sorry, what do I do? Do I go back? What?
Sir, okay lang po ba if kami na po ang mag-share ng presentation nyo? Meron po kaming copy of your slides. Hello. Hello po, Sir Guerrero. Shared na po ang inyong slides. I think muted po ang microphone niyo, sir. Excuse me, Sir Guerrero. Nakamute po ang microphone niya.
the interruption. Okay, can you hear me now?
Yes, sir. Okay na po.
Okay. My talk is on. My talk is on the application of tilapia sex reversal technology in the Philippines. This is quite a success story. It was developed at the Freshwater Aquaculture Center of the Central Luzon State University in the mid-1970s. Okay? Ayaw mag-move. Okay, let's start with the science or the scientific basis of this technology. In 1903, Bowen and Ansel proposed the hormone theory of sex differentiation in vertebrates, including fishes. The theory states that it is a sex hormone that triggers the development of the primordial sex cells, could be female or male, during the sexual differentiation stage, in the embryonic stage of the fish. Okay? Baldwin and Goldin in 1940 suggested that the administration of androgen, that's the male hormone, at the time of gonadal or sex differentiation may cause the generation of female germ cells. So if you introduce the male hormone to the genotype or the female developing its gonads like ovary, you can change the direction of the sex from the female to the male. Okay. This results in the conversion of primordial germ cells into male germ cells. Okay. Next slide. Okay. Okay. Yamamoto in 1958 reported for the first time the artificial sex reversal of a genotypic female. Now, genotypic refers to the genes, okay, of the medaka, a rice paddy fish in Japan, scientifically known as the Oricias latipes, using a synthetic androgen, or we call it also heteroleucos. It's not the natural hormone. Methyl testosterone. It's used in human medicine. No? I think for for curing cancer, I think, of the breast in human beings. Yeah. Clemens and Inslee in 1958 achieved the first functional sex reversal of female Tilapia mozambica, which is now Oleochromis mozambicus with methyl testosterone. Yeah. This was in 1958. Next slide. So,
Excuse me, Dr. Guerrero, naka-mute po ang inyong microphone.
yeah, okay. It's unmuted again. So normal, during the stage of sex differentiation, the female direction for female or phenotype. Now, phenotype is the secondary sex character, and the result is the sex organ of the fish will become ovaries. This is normal if natural process takes place. No? Female hormone is released, developing the primordial germ cells for femaleness. Right? For the male, it's the male hormone, natural male hormone, that does its effect, resulting in males that are phenotypic or functional males with testes as gonads. Right? However, with artificial sex reversal, the XX genotype remains the same. There's no change in the genes, but the sex of the fish is now directed towards maleness instead of femaleness, or phenotype. No, phenotype is the external characteristic, with a testis instead of an ovary. That's why we call it the sex reversal. Yung sex ng isda nababago. Okay? Imbis na maging babae siya, nagiging lalaki sa labas, pero ang genes niya hindi apektado. Now, with the male, there is no effect because it's the same effect because male hormone din ang binigay natin. So the phenotype of the male is that for testis, gonad. Okay? So I repeat, there is no genetic change in the animal, only the phenotypic or the what we call secondary characteristic. Okay. 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 Yamamoto in 1969 said that there are two conditions for sex reversal in feces to take place. The first is the heterologous, like methyl testosterone sex hormone, should be orally administered. You see, you cannot apply the natural hormone through the feed because it will be destroyed by the enzymes or stomach acid. All right. So these synthetic hormones can resist that, cannot affect it. So that's why it's easier to treat the fish through the feed rather than injecting them. You know, at that stage they're so small, the fry. So they are treated, administered orally, starting at the age. Now this is very important. You must apply the treatment before the sex differentiation stage. You have to establish that in the species. Okay, I'll tell you later on in tilapia when that takes place. We also call it the sexless stage, and continued throughout the stage of sex differentiation. If you treat it during the stage of sex differentiation, it doesn't work. And you have to do it for the duration of the sex differentiation and even prior to that. The second condition is that there should be adequate levels of hormones that should be used. Kasi kung kulang yung dose mo or yung concentration mo, it will not work. If you put too much of it, it will not also work. Okay, so you must apply the right amount or concentration of the hormone. Okay. Okay. Yun. Okay, in 1975, when I did my dissertation over, I'm glad to know that Dr. Oden is also a graduate of the same university where I studied. War Eagle, Dr. Oden, okay, produced all male offspring of tilapia aurea, or the blue tilapia, or Euchromis aureus, through sex reversal using the synthetic hormones ethinyl testosterone and methyl testosterone. If you're interested to receive a copy of this paper, let me know through email and I'll send you one. Okay. Then in the Philippines in 1976, Dr. Terry Abella and myself, by the way, Dr. Abella became the president of CLSU. He's now in retirement like me. Right? We induced for the first time sex reversal of the tilapia nilotica, the Euchromis niloticus with methyl testosterone. Euchromis, rather the tilapias then were not considered cultivable or not ideal for culture because they did not grow big, you know, although it was better looking, white, because of overpopulation, which I will explain later. Okay, next slide. Okay, I'm still mute.
Excuse me, sir. Naka-mute po ang inyong microphone.
Because my eyes are focused on the screen. I have to look down. Thank you for that. So for the first time, we produce large-sized fish. You know, if we don't control the breeding of the fish, they will just become stunted. Hindi lumalaki, small size. So considered yung tilapia noon peste. So it became attractive now to culturists because they can grow big, faster, and more marketable. Okay, kasi mas malaking price yung isda pag malaki. Okay. All right, so let's something is wrong, no? How do I move the slide? I'm muted again.
Excuse me, sir. Naka-mute po kayo ulit.
Ganun, no? Why do we apply it? Because the tilapia,
Sorry po for the interruption. I think Dr. Guerrero was disconnected. Let's just wait for him to reconnect. So let us use this time to acknowledge some of our participants. We acknowledge the participation of LGU Botolan, Municipal Agricultural Office, the LGU Mayantok, LGU Palongpon, the IGU Subic, Lions Club, LSPU LSC, the University of the Philippines Visayas, Lyceum of the Philippines University Laguna, Mabalacat City College, Pangasinan State University Binmalay Campus, Mariano Marcos State University, the BIFAR Regional Office 1, Marinduque State College School of Fisheries, Mary Hill College Incorporated, NFRDI, the Mindoro State University Bungabong Campus, the DepEd, Department of Education, NFRDI, Surigao State University Bungabong Campus, Surigao State
connection
College
here.
Dr. Guerrero is now reconnected.
Sorry. I have to move my slide. How do I do it? Yun. Okay. Tapos na tayo dyan. Sandali na lang ito. Okay. So let's review the technology, why we apply it, because the fish is early maturing, anak ng anak. So nagiging overpopulated. So what they need is population control. Because if you don't do that, they get stunted and you only harvest small size fish. By the way, in these species, the male grows faster and bigger. So if you have all male, you can produce more fish of larger size and more marketable. Okay, so yun ang basis or reason why we apply it. Okay, let's find out how we do it. Okay, first you must produce the sexually undifferentiated fry, which are 9 to 11 millimeters in total length. Now remember that, no? They're about 10 to 12 days old from post-hatching eggs in the hatchery nursery techniques. There are many hatcheries in the country, and they can produce the right fry at the right stage for treatment. Now you can produce them in pans, in tanks, or in hapas in pans, okay? Outdoors, yeah. Okay, next. And you feed the hormone feed. It's so simple. You can mix the hormone with the feed, dissolved in alcohol and so on. If you want more details, let me know. But this is done now by many hatcheries in the country. Concentration of the hormone is 30 to 60 milligrams per kilogram, or that's about 30 to 60 ppm. Okay, I'm unmuted, right? Okay, for at least 3 weeks. Again, you have to apply it 3 weeks, not 2 weeks or 1 week, because if you do it lesser time, you won't get the result you need. Or until 18 to 21. So they become fingerlings when they're about this size, no? Total length in outdoor or tanks, hapas, or pans. So it's quite simple. Rothbard and his colleagues were demonstrated for the first time using this technology, SRT, in 1983 in Israel for the mass production of hormonally sex-reversed all-male tilapias, Oreochromis niloticus, hybridized with Oreochromis aureus using ethinyl testosterone in covered outdoor tanks. I think they were able to get up to 100% sex reversal or males, no, in the population. Okay, being better and unmuted. Then Guerrero and Guerrero in 1988. Guerrero, the other Guerrero, by the way, is my wife, Luz Biminda, demonstrated the feasibility of commercial production of sex reversed Nile tilapia using methyl testosterone with open concrete tanks, resulting in 99 male fish. Now remember, in a normal mixed sex population, you have about 50-50, no? So you're only changing the functional sex of the females. No? The other 50%, the normal males, are not affected. Okay? Okay, what has this technology done or given us? In 1986 to 1988, the Philippines was the world's top producer, I think for the first time, no? Because there was no market for tilapia at that time. And because of the problem I said earlier, we produced 55,000 to 819 to 75,000 046 metric tons, according to Pulin in 1996. Tilapia became the second most important food fish in the country, next to milkfish. More than 50% of the Nile tilapia now produced in the world is believed to be sex reversed, so it's now applied in other countries. In 2019, more than 6 million metric tons of Nile tilapia were produced in the 80 countries or more than. Tilapia is now one of the top 10 fishes produced in the world today in terms of volume. It's also the second most important cultured fish in the world, next to carps. Okay. Let me answer some of frequently asked questions or FAQs. First is, what is the cost of producing? Is it expensive or hard or costly? It only takes 1 to 5 centavos more than that for the non-sex reversed fingerling. So napakamura. Okay? Depends on who's using it, rather applying it. No? Then the other question is it safe to eat sex reversed tilapia? Nakaka-cancer ba yan? O what? No? O baka nakaka-affect sa mga babae, nagiging lalaki? Hindi naman. So, ang joke nga dyan baka may bigote yung misis mo pag kumakain. So, sorry, that's not true. Okay. Okay, through radioimmunoassay studies, I think this is only one term, radioimmunoassay studies, okay, have shown that there are no methyl testosterone residues in the blood of Nile tilapia after 5 days from withdrawal. Remember, we eat the fish 4 months after we harvest them from stocking. Now we only treat the fry for 3 weeks for sex reversal. So 5 days after you stop the treatment, wala nang residue. So we don't eat the fry anyway. See it? Okay. The other question is, what happens to the metabolites? The concern is environmental naman. Baka yung mga waste products ng isda sa labas, makaka-affect sa environment, affect other species. Well, not like the birth control pills that women take that is flushed into the lake. It can make the what do you call this? The male fish become feminized in Laguna Divide. There's a study there, but it's not happening with this fish, no? With this technology, the metabolites that are thrown out into the lake or the outside do not affect other fishes because they are biodegraded or photolyzed or photooxidized rather. Nasisira ng araw tsaka ng mga bacteria. From a study done by Conklin and his colleagues in 2009. Okay, so now I come to the close of my talk and thank everyone for their patience and interest in this technology. Mabuhay! Okay, that ends my presentation. I will now escape. Okay, then what is next? Ano bang sunod dyan?
Thank you very much.
Yeah.
Thank you very much.
Nakamute na naman ako. Natapos din after all those disruptions. Anyway, thank you for inviting me again and for listening. No? I'll wait for your questions if there are any. Salamat po.
Thank you very much, Dr.
After speech.
Guerrero. Thank you, Doc, for sharing
Yeah,
your
sure,
knowledge
and
and
you're
for,
welcome.
for
You're
introducing the
welcome. Yes,
technology
yes.
of
Thank you.
tilapia sex reversal here in the Philippines.
Yes. Stop sharing pala. Okay. Can go back to Zoom.
Much,
After
Dr.
speech.
Guerrero.
Yan. Okay. Okay na.
Yes, Doc. Thank you po.
Welcome. You're welcome. Give my best to Dr. Kas. I can still see Ramji. Ramji, how are
Yes, Doc,
you?
we will po.
Yes, hi, Dr. Rafi, War Eagle. I'm very, very delighted to be sharing this webinar with you.
For extending their time to share their innovations to our participants.
And now, let us proceed to our open forum. May I request Dr. Mutia, Dr. Udin, and Dr. Guerrero to please turn on your camera and microphone for our open forum.
Hello.
If you still have your questions, you can still submit it to our Q&A box. The first question is for Dr. Mucha. The question is from an anonymous attendee. The question is, have you tried any other way to check fecundity, such as gonadal egg count? What is the maximum egg capacity for a 5-kilogram broodfish?
Okay. Okay. Thank you for that question. Yes, we tried counting the fecundity, gonadal egg count. So for a 1 gram, we have for 1 gram, we have 3,500 eggs per gram of the per breeder. Okay. So if we have a 200 to 500, we have a 200 to 500 gram per breeder of eggs. 200 the what you call the ovary, no? So for a so in that case, if we have a 5-kilogram broodfish, we can have a 1.1 million eggs. Yes. So we counted the eggs, at least 3,500 eggs per gram. And then the ovary, the gonad per breeder is around 200 to 500 gram per breeder. So if we have a 5-kilo broodfish, we can, for this 200 to 500 gram, we multiply the 3,000, no? So around 700,000 to 1.75 to 700,000 eggs to 1.75 million eggs per breeder. So for 5 kilogram of broodfish, we have around 1.1 billion eggs. So it coincides with my presentation earlier, no? That's around 223,000 eggs per kilo of breeder. Did I answer it right? Nalito ba kayo? Is that okay?
Thank you for your comprehensive answer, Ma'am Dr. Mutia. And for the next question, this is addressed to Dr. Udin and Dr. Mutia. And the question is, can we use xenogenesis in Maliputo or the other species?
Can we use what?
Xenogenesis po in Maliputo and or other species po.
I think
Hi,
Dr. Odin can answer that.
yes, thank you, Dr. Mutia. Yes, Anjali, to that question, I believe that there's a huge promise to and potential for the use of xenogenesis for all other species, including the maliputo. Basically, the promise of the genesis technology is for the reproduction of and to aid on the reproductively difficult to spawn species. So, you can actually develop a maliputo, or if there's a challenge on the production of maliputo, then we can potentially make use of xenogenesis and explore on how we could develop a protocol to develop a xenogenic maliputo that would be able to produce a better performing maliputo. But it really depends on how and on how on the target goal that we want to achieve. But yes, to the question, there is an immense potential and promise in terms of the use of xenogenesis in advancing biotechnology in aquaculture across all species. This has been done with all other species, including those other non-fish species. I believe that there was a study then that was conducted where a frog stem cells was isolated and then transferred into a fish species, and the fish species was able to produce a gamete coming from a gamete of a frog. And in other species also, in tilapia, it can actually be done. One thing that I am looking into here is for high valued marine species that are reproductively difficult to spawn, we can actually make use of xenogenesis in order to produce these high value species year round. We just have to identify the best and the most fit candidate as surrogate for the xenogenesis production of that species. Thank you.
Thank you for that excellent answer, Dr. Udin.
This next question is for Dr. Guerrero. Are there studies in sex reversal in milkfish, catfish, and other cultivable species? Sir, your microphone is muted po.
Okay, it has also been applied on the common carp, but not commercially. So far, tilapias are the only ones being used for sex reversal in the world. No, but there may be others, but they're still experimental. Thank you.
Thank you, Dr. Guerrero.
I don't think it is being applied in milkfish, no? Kasi lumalaki yung bangus eh. Hindi naman nagiging mature yan at saka nanganganak sa ponds. So like tilapia, they mature at about 2 to 3 months, and they breed every month. The milkfish matures in about 5 years. Tagal.
Thank you, Dr. Guerrero, for that comprehensive answer. The next question is addressed to Dr. Guerrero again. And the question is, sorry. Again, the question is addressed to Dr. Mutia, and the question is the technology introduced and adopted by the community? How about the terms of feasibility? Maris?
Yes. Thank you very much again for that question. Technology introduced and adopted by the community. How about in terms of feasibility? Okay, for the grow out culture, even before we started the breeding program, people are already culturing that in cage, so it's already existing. That's the reason why we try to breed them in captivity so that we can will increase supply of fingerlings for the grow out culture, because most of them are from the wild caught. So when we started this breeding program, of course, we also introduced the refinement on the grow out culture. So for the grow out culture, I think we started last year because during pandemic we need to provide livelihood also to our fisher folk. So from 2020 to 21, we have cooperator beneficiaries wherein we provide them inputs for cages and fingerlings. So I think I'll just answer. There's a question also in the on the feasibility. So for the information of everyone, for that project, the grow out culture for a 4 by 4 by 4 square meter or 64 cubic meter, we the common practice there is that the size of the cage, and they culture about 400 pieces of maliputo. Their survival rate is already quite good, about 48 to 90% survival rate, with a culture period of 150 to 220 days. So in 2 years, they have two cycles. So they are earning about 15,000 to 35,000 pesos per culture period for that small cage. So if they can multiply that cage, they can earn more. So next year we'll also do the same. So that is for the grow out culture. They are also doing a big grow out cage culture in Taal Lake. So it's about 10 by 10 cage size. So, but also the limitation is the supply of fingerlings. For the hatchery, we also try to collaborate with private hatcheries. They are very willing. We already tried with the big hatcheries in Quezon, in Pangasinan, in Zambales. But we are competing with the milkfish production. So, mas priority nila that time kasi pareho ng season yung maliputo and bangus. So, but we are still hoping in the next coming years that we'll be able to collaborate again with these private hatcheries when it comes to hatchery operations. Okay, thank you.
Thank you, Dr. Mucha. Our next question is for Dr. Odin. Sir, may I ask, what is the best strategy to increase the sperm count of a catfish? What is the best strategy if the catfish is already mature?
Hi, good morning again. I believe that the best strategy to increase the sperm count of the catfish is really on the broodstock management. That includes the feeds that you provide them, the environment that you provide them. It's a holistic approach that is needed in order to make sure that the reproductive performance of the catfish would be optimized. And what's the other question, Benjamin?
Recording stopped.
Is already mature?
Yeah, well, what was the question, Benjamin?
Also for the increase of the sperm count if the catfish is already mature?
Yes, in terms of there are multiple factors that would affect and contribute to the increase or to the number of the sperm counts in catfish. I believe that the
Recording in progress.
The genotype of the strain of the catfish also contributes to that. The genotype of the catfish, the environment provided for during the broodstock management or during the conditioning. This all contributes to the increase of the sperm counts. I think that in the case of the native catfish in the Philippines, if he's referring to that, there are very few academics or researchers working on the native catfish in the Philippines. There is a there, which is also an opportunity for other researchers in the fisheries to look into and investing on the native catfish and making use of biotechnological approaches, as well as optimizing the conventional approaches that we've been using for other species of catfish to make sure that we could boost the production again of the native catfish in the Philippines. But yeah, overall, going back, if we want to increase the production of the sperm cell, it should be holistic. That includes the broodstock management, including the feeds, the environment, and as well as considering also the genotype of the the genotype, the strain, the family of the catfish being used for that purpose. Thank you.
Very well said, Dr. Udin. And now for the next question. This is addressed to Dr. Guerrero, and the question is, aside from methyl testosterone, what is the most promising economically alternative hormone used for sex reversal?
Okay, but there are other ways to do it. In CLSU, they have done experiments using pine pollen. No? They got it from Baguio, yung pollen, yun ang mga ano ba yata, male, ano ba tawag doon, male seeds or gamits ng pine tree, right? Because it contains a testosterone-like compound, no, that has sex reversal effects. Although they get lower sex reversal, like about 90 to 95, no? We need about 90 you can contact the researchers in CLSU, I think Freshwater Aquaculture Center, about this. The French have also applied heat treatment. No? I think they raised the temperature to about 32 degrees centigrade for a certain period, and they can get about 70% sex reversal. So there is a temperature effect. No need for hormones, because in Europe, they do not allow the use of synthetic hormones for treatment of fish. Yeah. So those are two, and maybe others, no? But you'll have to go to the literature to look at that or look into it. Thank you.
Dr. Mucha, it was mentioned that photoperiod is a key factor in spawning as well as seasons. What is the photoperiod that you observe, and what is the optimal photoperiod used for maliputo?
Yeah, thank you again for that question. For maliputo, we do not regulate the photo period. We just follow the when it comes to injection and spawning, we just use the natural light, but we follow a certain timing during injection. So we start our injection at 6 a.m. in the morning for the first injection, and then in the afternoon it's 6 p.m. So, and then after that second injection, so medyo madilim na siya, we just place them in their hatching tanks. So we just allow them to spawn there. So we do not give them light. But in our larval rearing, we regulate the light. So it's 16 hours with light and 8 hours dark. So that means around when we say 16 hours, it's from 6 a.m. to 10 p.m. we turn on the light in our indoor hatchery. And then we turn off the light from 10 p.m. to 6 a.m. So that is proven on our part, but I think we have to do it again to refine it. But for now, it's okay naman yung larval rearing namin if we control that timing, lighting, photoperiod. But for spawning, no, we don't manipulate the light, but we just follow the timing. Okay, that's all.
Thank you po, Dr. Mucha.
Dr. Maris, I have a follow-up question for you, ma'am. How did you come up with using LHRH and HCG? Did you refer to the reproduction cycle or spawning of other cultured fish species? Also, Maliputo is a seasonal spawner po, ma'am.
Okay. Parang may nalimutan ako kanina dun sa ano, kasi she's talking about yung season, no? Dun sa photo period. Am I right? You talk about season. Well, for season, we follow the new moon and the first moon during our injection. Okay. So for your question on how did you come up of using LH and ACG, no? Of course, we look for references, especially for marine fin fishes. So we look on the references for breeding sea bass, milkfish, snapper, grouper, and we tried the different hormones that they are also using. So it's not only LHHCG, we also use the CPE, the crude pituitary extract. But among the three, the LH is more feasible to us. So we get higher fertilization and hatching rate and successful spawning when we use the LH. Yeah. Did I answer it right?
Yes po, ma'am. Thank you po, Dr. Mucha. And unfortunately, due to limited time, this is our last question for our open forum. And this question is addressed to Dr. Guerrero. Dr. Guerrero, I'm curious of the age of the tilapia seedling that undergoes sex reversal using MT. What is the best age of the tilapia seedlings for that undertakings?
Well, I think I gave that earlier. So the best age is, well, for most of the tilapias we raise, like the Mozambicus and the Aureus and the Niloticus, the best.
Sinasagot mo ba?
We're very sorry po for the interruption. I think Dr. Guerrero was disconnected again. Let's just wait for him to reconnect po.
Feed them with the hormone feed, yung feed na may concentration ng methyl testosterone. Or you can wait till the females that are mouth brooding to release them into the water. You collect them. So that's about the right stage. That's about 11 millimeters long or 10 days from post-hatching. So importante yung timing mo. Do it before sexual differentiation that takes place within 3 weeks after the fry are released by the female or when they are about 11 millimeters long. Okay.
Again, thank you for answering those those questions and for the great presentations. It was a pleasure to have you with us today, Dr. Mutia, Dr. Udin, and Dr. Guerrero.
Once again, thank you to all of our dear speakers. Now we will award the Certificate of Appreciation to our speakers. This Certificate of Appreciation is awarded to Dr. Maria Teresa M. Lucha for sharing her valuable time as plenary speaker during the 5th National Fisheries Biotechnology Symposium with the theme Advances and Innovations on Reproductive Biotechnology in Aquaculture and Fisheries on September 29, 30, 2021 via Zoom, given this 30th day of September 2021, signed Dr. Lillian C. Garcia, CESO 5, Acting Executive Director of DA and FRDI. The same certificate will also be awarded to Dr. Ram G. Y. Odin and also to Dr. Rafael D. Guerrero III. Once again, thank you to our speakers for gracing the second day plenary session with your expertise.
At this moment, may I request everyone, including our speakers, to please turn on your camera for our photo opportunity. And the count of 3, give us your best smile. Okay. Are you ready? 1, 2, 3, smile. Again po. 1, 2, 3, smile. Okay. Thank you very much.
Thank you.
Thank you everyone for bringing their expertise, experience around the table, engaging in such a fruitful, constructive, and open exchange throughout these 2 days 5th National Fisheries Biotechnology Symposium.
Thank you.
So at this moment, let us entertain by Mr. Lawrence I. Norte.
This is the moment, this is the day when I send all my doubts and demons their way. Every endeavor I have made ever, it's coming into play, it's here and now today. This is the moment, this is the time when the momentum and the moment are in a rhyme. Give me this moment, this precious chance. I'll gather up my past and make some sense at last. This is the moment when all are done, when all the dreaming, scheming, and screaming become one. This is the day to sparkle and shine, when all I live for becomes mine. For all this year I've faced the world alone, and now the time has come to prove to them I make it on my own. This is the moment, my final best. Destiny beckoned, I never reckon second best. I was the moment, the greatest moment of them all. This is the
Close this webinar. Now, to formally close this webinar, we are pleased to have our ever-supportive head of the NFRDI Fisheries Biotechnology Center, Dr. Gasciano H. Teresca Jr. Let us all give him a virtual round of applause.
Hello, good morning. Good morning po sa ating lahat. So it's an overwhelming NFBS. No? It's a 5th NFBS. And I think we have around 400 plus attendees. And I think we have 1,000 plus registrants all over the country. And I think we have attendees from abroad. No? Marami tayong mga participants from abroad. And I think this is a well-anticipated event every year. And actually, this is just a build-up activity of our NFBS. And looking forward this November for our 6th NFBS na ang topic po natin ay advances on aquatic animal health. So this one is on advances in reproductive biotechnologies, in aquaculture and fisheries, and the topics are overwhelming. It's an going international na talaga tayo sa fisheries. So napakagandang mga topics, no? And never heard before about xenogenesis, induced breeding on marine fish. And of course, our maliputo na nandun po sa pera ng natin, no? And sex reversal in tilapia. I think this is the first, no, the father of sex reversal, no, in tilapia is here in the country in the person of Dr. Rafi Guerrero. So, sir, thank you very much for this opportunity for yung isang isang tawag, isang text na paunlakan niyo po kami dito sa Fisheries Biotech, dito po sa NFRDI. And really indeed, networking works, no? It's really it's really a network of researchers and researchers, and of course a network of friends. So first of all, thank you very much to our great speakers for the first day, like in day one. We have on groupers in the person of Sir Peter Palma from CIFDIC AQD. So great presentations on commodity on groupers, no, mga high-valued species natin. Makikita po natin, pwede pala natin ma-induce yung maturation ng mga groupers. And we have this young researchers now na nagwo-work on aquatic biotechnology, and it's really promising. And we have on chub mackerel. So yung malay mo, mapapaanak na natin yung tuna dito sa Pilipinas, and we have vast security, no, na maipapaanak natin. So in the person of Dr. Sunny Lumayno from University of the Philippines in the Visayas. And we have import from Turkey. Of course, our friends in Dr. Rami. So technologies that we have, no, gagamitin natin sa aquatic and fisheries biotechnology. And today, overwhelming yung participants na hindi talaga lumabas sa ating Zoom link ngayon, though we have difficulties in achieving the internet connection, but still our participants is still now around 400 plus. And it's, pride talaga ng NFRDI to share in the person of Dr. Maris. Thank you, Ma'am Maris, for sharing the Maliputus story in the industry. And of course, we have import also from Mindanao State University, Maguindanao. Sir Ramji, sorry for yung mabilis lang pag-text sa'yo. For I think a week lang nasabi namin, Sir Ramji, we need your help to present on xenogenesis. And right after, it is just a text away that you responded very quick for our message, and you are there. So thank you very much, and my regards to the MSU family there out there in Mindanao. So very overwhelming that meron palang mga works on xenogenesis in the catfish, and we can improve our catfish in the Philippines. And catfish now is a booming industry, not only here in Luzon, but I think in Visayas and in Mindanao. And through this platform, we can help the fisheries industry through biotechnology. So hindi yung parang ang ano natin kasi ng biotech is just like highfalutin word, and yet instead, meron na palang nag-i-exist nito. Like for example, the sex reversal of tilapia, which na-introduce sa atin ni Sir, na kung ako mag-isip, parang hindi pa ako Sir Rafi na ipanganak yung 1970s, late 70s na. But we have that already. So the first pala yung tilapia, yung transgender yung tilapia na nagagawa na. So overwhelming works of science, and of course, in the field of aquaculture and fisheries biotechnology. And sa ating mga speakers, maraming salamat po sa pagpaunlak. Napakagaling na presentations this morning and yesterday. And overwhelming po kami na pinaunlakan niyo yung aming invitasyon. I hope we can have that again next year. And we are hoping next year to introduce the cell transplantation and of course, cloning or nuclear transfer in fish. So abangan po natin, and there are lots of benefit fish species in the country and of course in the world na pwede nating gawing laruan na ma-improve po natin ang aquatic or reproductive biotechnologies. And this is not new. This is just mga promotions natin. And if we discover this one, and I think we have a lot of students right now to engage in reproductive biotechnologies. Engage in research, engage in new technologies. No? Hindi ito bago. It's a matter of acceptance na magiging i-accept natin yung mga bagong technologies sa ngayon and i-apply natin in fisheries and aquaculture. And of course, maraming salamat po sa aming mga boss, sa mga supervisors po natin, kay Ma'am Lilian Garcia, ang Acting Executive Director ng INI4DI, yung colleagues po natin sa INI4DI. Nakikita po dito, Ma'am Elsa. Hello po, Ma'am Elsa is our former chief dito sa Fisheries Biotechnology Center. And of course, sa mga colleagues po natin, ng mga taga-INI4DI, and of course, sa DA Biotech Program na nag-implement at nagtulong po sa atin na mapunduhan itong ating mga projects dito po sa Fisheries Biotech Center. And of course, with the support of Ma'am Jodicel Danting, the Center Chief of the BFAR NFFTC. So dito po tayo na in-house po and in collaborations po yung mga project po natin dito kay Ma'am Jude at sa mga taga BFAR NFFTC attendees po natin, maraming salamat po sa pagsuporta po ninyo sa amin. And of course, our networks of sabi na natin, network of friends and collaborators from state universities and colleges, of course, from the academe, research institutions, and yung mga kasama natin sa industry, stakeholders, mga students, maraming salamat po. And I hope we impart you this very momentous and of course this productive scientific symposium in the field of aquaculture. Thank you din po sa ating mga FBC staff na puyat, no? At nagiging dito yung sa atin, nagiging instrument na mapatuloy natin itong ating symposium. Kasi yung internet connection ngayon napakahina dito. So with regards with that, napakagaling na naanhan natin na overcome po natin. Marami po tayong mga networks dito na nakakamit para lang maipatuloy natin ang ating symposium. And very, very thankful sa inyong lahat, sa inyong suporta. And of course, next year or this year, maraming programa po ang FBC and it is for free. Abangan po natin doon sa ating mga website and we are happy to share this opportunities sa ating lahat. And with that, maraming salamat po sa inyong sa pag-attend po ninyo and don't forget to yung mag-read sa link para sa inyong sa e-certificates. And that's all. And magandang umaga po. Happy lunch po later sa inyong lahat. And regards po sa inyong mga institutions. That's all po. Maraming salamat.
Thank you, Dr. Cheresca, for that motivational remark. Again, thank you to all the speakers and thank you everyone for attending this today's webinar, the 5th National Fisheries Biotechnology Symposium. On behalf of NFRDI Fisheries Biotechnology Center, thank you for joining us today. Mabuhay ang biotechnolohiya. Stay safe and God bless us all.
This is the moment.
Thank you po. Mama Riz, thank you. Doc Ramji.
Bye.
Thank you. Si Rafi.
Thank you, Doc Cas.
Thank you, Doc.
Thank you po sa lahat.
Thank you, everyone.
Ma'am Ju. Doc Ramji, regards sa MSU.
Thank you po.
Yes, Doc Cas, thank you. In the regards.
Here and now today. This is the moment. This is the time.
Thank you, everyone. We would like to acknowledge the participation of Colegio San Agustin Bacolod, College of Fisheries Central Luzon State University, College of Fisheries and Aquatic Sciences, College of Fisheries DSB CKB, Kawarda Chhattisgarh, India, Cotabato Foundation College of Science and Technology, Cotabato State University, the CSU Apari Campus, CSUA, DANFRDI, and BIFAR NIF-EDC, The Touch Aqua Farming Incorporated, Davao del Norte State College, De La Salle Lipa, De La Salle University, Manila, Department of Agriculture, Department of Agriculture, Bureau of Fisheries and Aquatic Resources, the Department of Education, Department of Environmental Science, the Department of Fisheries and Aquaculture, UGAS, Department of Interior and Local Government, Department of Science and Technology, Region 12, Department of Education, Legal Fisheries School, Department of Education, Alcala Rural School, Department of Education, Kabatuan, NCHS, DHBSU, DILG, Don Honorio Ventura State University, Don Mariano Marcos Memorial State University, the DOST, the Eastern Summer State University, the Emperor Gardo Cabo Pontevedra Capiz, the Faculty of Aquaculture and Fisheries, Far Eastern University, Manila, the Feedmates Specialist Incorporated Group, Fisheries Biotechnology Center, Francisco E. Barsagas Integrated High School, GMPCC Siruma, Governor Mariano E. Villaferte Community College, Siruma, Robets Feeds Philippines Incorporated, Aras State College, the Buia Aquaculture, Awasa University, ICAR National Institute of Abiotic Stress Management, Ilocos Sur Polytechnic State College, the Iloilo Doctors College, the Iloilo Provincial Agriculture, the Iloilo State College of Fisheries, the Industrial Technology Development Institute, the Institute of Aquatic and Applied Sciences, the Institute of Fisheries Postgraduate Studies in Iraq, Isabela State University, JDEX Farm and Hatchery, the JMA Aqua Farm, Calibo Save the Mangrove Association, Castamono University, Kerala University of Fisheries and Ocean Studies, the Laguna State Polytechnic University, Land Bank, and Leyte Normal University. Thank you so much for your participation.
Also, we would like to acknowledge our participants from University of Santo Tomas, Carlos Hilado Memorial State College, Binalbagan, Iloilo State College of Fisheries, Northern Negros State College of Science and Technology, University of Southern Mindanao, University of the East, University of the Philippines, Manila, MSU Maguindanao, Zamboanga State College of Marine Sciences and Technology, LGU Bayan City, LGU Binian City, Batangas State University, LGU Borongan, LGU San Pedro, LGU Sapiantati. Also, we have from Pangasinan State University, Ministry of Agriculture, Fisheries and Agrarian Reform, Pangasinan State University, Polytechnic University of the Philippines, BIFA Region 1, PRMSU Candelaria, Provincial Agricultural Office, Romblon State University, Ragay National Agricultural and Fisheries School, Rizal Technological University, Bono, Rizal Technological University, College of Arts and Sciences. We have Rapid Mix Specialist INC2. Thank you, everyone.
Destiny beckoned, I never reckoned second best. I won't look down, I must not fall. This is the moment, the sweetest moment of them all. This is the moment that won't be ours. Today and never, I'll be forever with my God. When I look back, I will always recall moment on moment.
Again, our evaluation link is already posted in our chat box.
The greatest moment
Evaluate before the deadline. Thank you.
of them all.
It is also available in our Facebook page and YouTube.
This is the moment. This is the day when I send all my doubts and demons their way. Every endeavor I have made ever, it's coming into play. It's here and now today. This is the moment. This is the time.
For more updates,
when
follow
the
us at
momentum
Fisheries.Biotech Facebook page and visit our website at fcc.nfbi.ca.gov.ph. For more updates on our activities here at NFBI Fisheries
I'll gather up my past and make some sense at last. This is the moment when all I've done, when all the dreaming, scheming and screaming become one. This is the day to sparkle and shine, when all I live for becomes mine. For all this year, I've faced This is the moment. This is the day when I send all my doubts and demons their way. Every endeavor I have made ever, it's coming into play. It's here and now today. This is the moment. This is the time when the momentum and the moment are in a rhyme. Give me this moment, this precious chance. I'll gather up my past and make some sense at last. This is the moment.